Hello guys, today we will discuss about homopolysaccharide, very important for biochemistry students. Homo mean same, poly mean many, saccharide mean sugar. It means the carbohydrate which are composed of more than 10 monomers of same repeating monosaccharide is known as homopolysaccharide. Once again, the carbohydrate which are composed of more than 10 monomers of same repeating monosaccharide are known as homopolysaccharide. For example, starch is a homopolysaccharide of glucose, glycogen is a homopolysaccharide of glucose. Similarly, cellulose is only made up of glucose. Then we have inulin. Inulin is composed of fructose monomers. Then we have chitin cell wall of fungi is composed of N-acetyl-glucosamine. N-acetyl-glucosamine. So homopolysaccharides are very important in biochemistry because they are the source of food. Almost all carbohydrates are initially started from starch. For example, glucose are obtained mostly from starch. Maltose, isomaltose mostly obtained from starch. So polysaccharides are more complex substances. Some are polymer of a single monosaccharide that term as homopolysaccharides, such as starch, glycogen. Some contain other group other than carbohydrates, such as hexuronic acid, hydronic acid are called as heteropolysaccharide, like mucopolysaccharide or glycosaminoglycan. Starch is a polymer of glucose that occur in many plants as a storage food, and it is stored in the leaf stem as well root, fruits, seeds, where it is usually present in greater concentration. Mostly, it is present in the grains, tubers. Potato tuber contain lot of starch. So mostly starch is composed of two units. One is mylose and other is myelopectin. Mylose only a state chain homopolysaccharide unit, and it occurs only 15 to 20% in whole starch, and its molecular weight around 60,000 Dalton. It is soluble in water because it has low molecular weight, give blue color with blue gold iodine solution. Structure unbranched mean it is straight chain. Why? Because it contains only alpha 1,4 glycosidic linkage. Alpha 1,4 glycosidic linkage made straight chain, but alpha 1,6 glycosidic linkage make a branch. So this alpha 1,6 glycosidic linkage is not present in mylose. It is only present in the myelopectin. Mostly mylose twist into a helix, and one turn contain around 6 residue of glucose. While myelopectin is more concentrated in starch, it is present around 80 to 85% of starch, high molecular weight around 5 lakh, insoluble in water, can absorb water and swell up, gives reddish violet color with blue gold iodine solution. Highly branched structure which contain alpha 1,6 and alpha 1,4 glycosidic. Alpha 1,4 glycosidic linkage is responsible for straight chain polymerization, but alpha 1,6 linkage which comes after the 24 to 30 residue of straight chain is alpha 1,6 glycosidic. So it is branch structure, but it is less branched as compared to glycogen. So there are two types of mylases which degrade starch. One is alpha mylase, which is present in saliva and pancreatic juice. This alpha mylase has random action. It can break any alpha 1 4 glycosidic linkage. But beta amylase has a specific action. It removes the maltose residue from non-reducing it. So it is specific. Beta amylase is specific. At the end, we have 6 to 10 residue glucose polysaccharide, which is known as limerdextrin. Limerdextrin cannot be destroyed by beta amylase. So here you can see state chain polymerization alpha 1,4 glycosidic linkages means all glucose in the alpha configuration. And when we have state chain polymerization of around 24 to 30 residue, then we have a branch. Then we have a branch of alpha 1,6 glycosidic. Uses of starch. Starch has a wide variety of application in food, drugs, industry. It is used as an antidote for iodine poisoning. Why? Because it can absorb iodine from your body. That's why it is used as an antidote for iodine poisoning. Diluent in powders and tablet manufacturing. It dilute the diluents. Nutrient, demulcent, protective and absorbent. Starting in the manufacture of glucose, liquid glucose, maltose, and dextrin. All glucose residue or maltose residue are obtained by the hydrolysis of starch. Glycogen is also known as animal starch and it is stored in the liver and the skeleton tissues. In higher animals, it deposited in the liver and muscle as a storage material which are readily available as immediate source of energy. It is dextrose. Formation of glycogen is known as glycogenesis, and it is triggered when we have a high carbohydrate meal. And breakdown of glycogen to form glucose is called glycogenolysis, and it occurs during starvation condition. Postmortem glycogenolysis is very rapid, but ceases when the pH falls to 5.5 due to lactic acid formed from glucose. So, initially in postmortem, it is the criteria to check the time of the death. It is high molecular weight, around 10 lakh to 40 lakh. Solubility glycogen is not readily soluble in water but form a pearlescent solution because it is not readily soluble in water. It can be precipitated from pearlescent solution by ethyl alcohol. If we add ethyl alcohol and then we dry it, it will make anhydrous powder, pure white powder. Action of glycogen is not destroyed by hard, strong alkalis like potassium hydroxide and sodium hydroxide. This property is made use of in the method for determining it quantitatively in tissue. We can quantify the level of glycogen in our tissues with the help of strong alkali. Why? Because strong alkali does not destroy glycogen. Action with iodine, glycogen give deep red color with the lugol iodine solution. In this respect, it is resemble with the erythrodextrin. Structure similar with the starch, but it is highly branched. In starch, branch comes after 24 to 30 residue, but here branch comes after 12 to 20 residue of glucose in a state chain. Here you can see this is state chain polymerization, then we have branch. This is state chain polymerization, then we have branch. This is state chain, then we have branch. So branch comes after 12 to 18 or 20 residue. Phenolene is a polymer of D-fructose and has low molecular weight, around 5,000 Dalton. It is the location of phenolene occur in the tubers of dahlia and roots of Jerusalem, artichoke, dandelion, and in the potato. It is also present in the leaves of onion. It is also present in garlic. It is liver rotating and give no color with iodine or light yellow color with iodine. Acid hydrolysis produce fructose. Similar, it is also hydrolyzed by inolinase enzyme, which is only present in the plants. It has no dietary importance in human because inolinase enzyme is absent in human. That's why it is also used as a laxative agent in humans. It has two most important functions in animal and the human body. It is used in the physiological investigation and determination of rate of glomerular filtration rate. So, why when we inject the inulin in blood, what happen? Our kidney filtrate. If kidney take a lot of time to filter it, then our kidney is not working well. It has been also used for the estimation of extracellular fluid volume. Other uses of inulin, it is used as a diagnostic agent for the evaluation of glomerular filtration rate. We have already discussed. Used as a soluble dietary fiber as a laxative agent. Used as a appetite suppressant. Why? Because it is not digested by human body. Used as a low glycemic index sweetener. Very good. Also used as a fat cream substitute. Cellulose, one of the most abundant organic compound in this universe. Cellulose is a polymer of glucose. It is not hydrolyzed directly by dilute acid, but heating with fairly high concentration of acid yield disaccharide of cellulose and glucose. It is composed of beta 1,4 glycosidic linkage. Why? Because all the glucose residue are in the beta configuration. Cellulose is very stable and soluble compound. Since it is the main constituent of the supporting tissue of plant, it forms considerable part of our vegetable food. And it is in food, it is act as a laxative agent. If you have constipation, then you have to use cellulosic diet. Herbivorous animals have bacteria in their lumen. That's why it can digest cellulose. So cellulose has a nutritive value in the ruminant animals, but it is of considerable human dietary value that it adds bulks to the intestine content, therefore stimulating the peristalsis and elimination of undigested food residue. So it acts as a cellulose act as a scavenger in animal body. Why? It can remove the undigested food particle which is present in the villi of small intestine and remove that toxic compound along with it. Macrocrystalline cellulose used as a binder disintegrant in the tablets. Methyl cellulose suspending agent and bulk laxative agent. Oxidized cellulose like hemostat is used in many applications. Sodium carboxymethyl cellulose is used as a laxative agent in constipation. Cellulose acetate, rayon, photographic filament plastic have many applications. Cellulose acetate phthalate is used in indirect coating. Then nitrocellulose is used in the explosive. It is also used in collodion. Dextrin. Dextrin is the hydrolyzed form of starch. Mean low molecular weight homopolysaccharide of starch is known as dextrin. It has alpha 1,4 glycosidic. The example starts by being precipitated by alcohol forming sticky gummy masses. Biomedical importance, dextrin solutions are often used as a mucilage agent. Mucilages mean which are used in the back of the stems. Starch hydrolysate consisting largely of dextrin and maltose are widely used in infant feeding. Why maltose is used in infant feeding? Because it is easily digested by amylase. Limit extent, it is well defined extent. This is the product remaining after the beta amylase and alpha amylase action has acted upon starch, but no further action is observed. Undigestible extent are developed as soluble fiber supplement for food products and used as a laxative agent. It is also used as a thickening agent in the food processing. Then we have dextrine. Dextrine is the polymer of glucose, and it is obtained uh, it is synthesized by the action of gluconostage mesenterite, which is a bacteria, non-pathogenic gram-positive Fukai bacteria, which use the sucrose as a growth medium. In sucrose, we add gluconostage mesenterite, where it utilizes fructose for its food and glucose for different linkages like alpha 1,4, alpha 1,3, and alpha 1,6 glycosidic linkage, and form a network to make dextrine. And dextrine has many uses. Why? Because it acts as a plasma expander. It has molecular weight around 75,000. When we inject it intravenously, so it acts as a blood. Why? Because it has low disintegration rate, low osmotic pressure, and long life in the blood. It increases the blood volume because of its high viscosity, low osmotic pressure, slow disintegration rate, and utilization, and slow elimination from the body. They remain in the blood for many hours to exert its effects. That's why it is used as a blood replacement therapy, because it has high life in blood. It has low osmotic pressure, low disintegrate, and high viscosity, which is very important for blood during hemorrhage condition. What is hemorrhage? When we lose a lot of blood during an accident. Only disadvantage is that it can interfere with grouping and cross matching as it forms fall agglutination. When we add this dextrine in our blood, what happen? It will make coating over the surface of blood, and our antigen will not exposed toward the antibody. That's why we cannot identify the blood group after the injection of this dextrine. Agar, which is very important homopolysaccharide, made up of repeated unit of galactose when it is, which is sulfated mostly at the carbon number 4. Present in seaweed, it is obtained from them. Biomedical importance, it acts as a laxative agent during constipation. Like cellulose, it is not digested, hence add bulk to the feces and help its propulsion, mean it remove the undigested food particle. In microbiology, agar is available in purified form. It is dissolved in the hot water. On cooling, it sets, mean it solidifies. So it is used in agar plate for culturing of bacteria. Why? In cold form, it sets in like solid form, but after heating it liquefy. If you like that video, please subscribe that channel. Thank you all. Team Manila. These are the references of different books where you can see that lecture.